Next up, everyone, we have Bioxytran Inc. They're in the OTCQB under the ticker symbol BIXT. The company is a clinical stage biotech company with two revolutionary platform technologies that were quickly developed using the power of AI. They have a broad spectrum oral antiviral drug that recently completed clinical trials and achieved 100% viral reduction in 7 days, a feat only accomplished by one other drug in the past decade. Their drug is a galectin inhibitor that not only neutralizes COVID-19, but also neutralizes influenza and RSV and prevents it from entering cells. They are currently recruiting patients for their dose optimization study, which will give them the ability to proceed with their registrational trial in India after they finish. They also have another platform drug being developed to treat strokes. Due to the molecule's small size, it has the ability to carry oxygen into areas of poor circulation. We want to welcome back Mike Sheikh. Nice to see you, Mike. Always happy to have you back. Welcome back to the conference.
Yeah, well, thank you. It's nice to be here.
Now, before we get into your presentation and the Q&A, I always learn so much from your presentations here. I want to talk about something pretty relevant.
Would you want to save that to after the presentation, Anna?
Okay, sure. Let's say that for after the presentation. So give the presentation and we'll come back with the Q&A and I'll mention my topic.
Okay, I love it. My company is Bioxytran. I'm the Chief Communications Officer. I'm pleased to be here to give this presentation about Bioxytran. And you'll see the symbol, by the way, is BIXT on OTC markets QB. It's AI-inspired drug design, optimizing carbohydrate drugs. So what we're about is, you'll see the tagline, we want to treat diseases instead of symptoms. Let's get to our forward-looking statements and knock them out of the way. You can read that at your leisure. Now, typically, this is the spot where we start with a mission statement, learn about the company. I just kind of want to dive in with our investment thesis so I can really capture your attention on why you really want to pay attention to what I'm about to tell you. This is the investment thesis. So first of all, Bioxytran has a game-changing platform technology. I'm going to tell you more about that later on. We have clinical trial results that validate the science, which ultimately means we're going to have a high probability of success moving forward. And it's not inappropriate to say that. And that's what you'll see when I point out that in the presentation. We also have IP and proprietary science, and the AI helps us figure out how to develop these carbohydrate drugs that neutralize viruses. And we're also going after chronic diseases. So the big picture is this is social impact investing that we're talking about. We want to make the world a better place. So again, that's part of this overarching investment thesis. And we have an extremely compressed valuation, which makes us at an ideal buying level. So again, we're going to show you in this presentation why that's the case and give evidence behind what the models suggest. And we have, in biotech, it's so important to have a solid management team. And we do. We have a solid management team, and we have a strategy to basically make you, the shareholders, money. And we can't wait to share that with you. And finally, for the OTC investors out there that care about this, there's a thin float, loyal shareholder base, and all that is the ingredients for a potential explosive upside. So with that, let's get to my trial data. Well, if you see on the left, what's missing? Graphs and charts. I'm not trying to confuse investors anymore. I just want you to look at one thing. So that page is left intentionally blank on purpose. If you want to see the clinical trial results, they're available on the website. Go for it. But I want to stress the reason I'm not showing this is I don't want to confuse you. I want to make it very well known. We had 100% responders rate in a clinical trial, and the p-value was 0.001. That's it. End of story. I shouldn't even need to give the presentation. You should just want to buy based on that, because any biotech that rises to that level of excellence, you want to pay attention to. I'm not saying buy the stock. I'm saying pay attention to what we have. So how did we get this? We had complete viral elimination of the viral load in 5 days. We reduced infectivity, we quieted the cytokine storm, and we had robust antibody response, which we nicknamed post-immunization, post-infection immunization. So with that, let's move on to the fact of what it is that we're selling. We're selling broad, or it's not selling, but what we're developing. We're developing broad spectrum drugs, specifically broad spectrum antiviral drugs. And what you'll see here is everyone's heard of broad spectrum antibiotics. Look at all these broad spectrum antibiotics and look at all the companies backing them. There's in this whole field of research of broad spectrum antibiotics started with penicillin. It was the first one that led to all the creation of all these antibiotics. And can you imagine a world without antibiotics? Think about a world without antibiotics. So I can't imagine a world without broad spectrum antivirals. So if you see there, page left intentionally blank. I did that again for impact. We believe that we hold the key to this area of the marketplace. And I wanted to show you this way so you can see how big it is. Now, why are we the key? Because all these guys on the top half are going to want to get into broad spectrum antivirals. And we hold the key because we have a patent pending with broad patent coverage on over 60 viruses. Think about how that could be a game changer. So again, I'm painting a picture of why you, the investor, needs to pay attention to this. As an investor, we believe that there is a huge valuation gap that you're going to be able to exploit. Now, how did this gap happen? Well, it happened as a result of the artificial intelligence that we were using in our NMR technology to identify compounds that work. We basically used NMR technology to see, does our molecule bind to the spike protein? And if it binds to the spike protein, it will prevent viral entry. So what says that we can't replicate this process for all the other viruses that are out there? That's how artificial intelligence has accelerated our pathway to development. We don't need to spend 5-7 years in that early stage trying to figure out what molecule works or doesn't work. We can do it very quickly with AI. Now, Baybridge Bio came up with this valuation model that you see there on the left-hand part of the slide. So where are we at? We are right at the big X, okay? So we should be at a 1.248, or we'll just call it 1.25 billion market cap valuation as a phase 3 ready company. Current market cap is kind of plotted on there, and there's a gap. It's a noticeable gap. Well, why is that gap? Well, because a lot of people don't realize what stage of development we are at, or how big this platform is. And that's what the rest of the presentation is going to really shine on. Now, the other thing that I'll point out with this slide is it shows, if you go to the right, it's the probability of success. It's 25% based on Baybridge Bio's model, okay, at this stage. Drugs that get to this stage have a 25% likelihood of approval at this stage, according to the model. So how can we leverage or increase the odds? Well, take that same molecule and use it in more disease indications is another way to increase the likelihood of approval. So that's another way where our technology is de-risking on itself if we do a label expansion. We're not a one shot in the dark. So now finally, I'm getting to, I overshot here. I'm getting to what our company is about. We make from complex carbohydrates drugs that are able to treat viral infections, cancer, and provide oxygen support. It's kind of a broad-based platform technology. And the guy behind this technology is none other than our CEO and CSO and chairman, Dr. David Platt. Dr. Platt is amazing in the sense that he coined the name galectin. You'll see that in 1992. So all this is a result of something that he started on his journey a long time ago. And that's what I really want to impress here. So when it comes to galectin science, this is the right person to lead the team and the charge forward. And you'll see his accolades, textbooks, public companies, journal articles, patents, clinical trials, lots of accolades. Now, what supports how big this technology is? And it says galectin-3, one receptor for an alphabet diseases from A to Z. Sorry that it's coming in kind of slow here, but you see for every part of the alphabet, if you look into the research, there's a journal article that suggests a disease indication is applicable. Now, these are big disease indications. Look, asthma, atopic dermatitis, these are billion-dollar indications. COPD, how about cancer? Diabetes, is that small or big? I mean, just heart, HIV, influenza, idiopathic arthritis, kidney, liver fibrosis, NASH, the largest unmet medical need, right? Let's go to the next slide and finish it off. Well, you can read this stuff. The idea is each letter of the alphabet is almost like $1 billion type of indication. And that is the takeaway that I want you to get. That's how big this has the potential to be. Once we get a breakthrough and approval in one indications, many more to follow. And it's important that investors see that potential so you know what you're investing in. And we're first and have the most not first, but we have the most experienced management team to take this to fruition. Glycovirology, a new science. What is glycovirology? Glycovirology, simply put, is a study of how carbohydrate drugs interact with viruses. So again, how carbohydrate drugs interact with viruses. So here's something that we found in glycovirology. There is some broad spectrum antiviral activity. You know, viruses can be broken down into DNA viruses and RNA viruses. And if you look at the RNA viruses, you see the second one, and you'll see down there influenza. And then let's go to the far right, paramoxiviridae. You know, that's a RSV. Right next to it, a deadly virus called Nipah virus. And then SARS-CoV-2. You know, so we have in vitro studies that support and our contention that it works in all these different viruses. So with broad spectrum antiviral activity. And for viruses, you go to the doctor's office, there's really, no matter what virus it is, there's no real effective treatments out there. And these data present a large market that we can go after. I mean, look at how many people got COVID-19 infections, influenza, shingles. These are like almost annual type cases. And there's no broad spectrum antiviral. Remember I left that page intentionally blank? Okay. There are no broad spectrum antivirals. Does anyone see that as a huge need? We're the key to unlocking that need. Or our technology is the key to unlocking that need. And that's what we're so excited about. Sorry, the graphics are loading slow on this, but we're the first line of defense against all mutations of coronavirus. So we have a COVID drug. It's versatile. It doesn't matter what variant you have. It's been tested. It's non-toxic, and you basically are PCR negative in 3 to 4 days. We're not expecting any limitations, whether you're pregnant or old or have any underlying medical conditions. We're not expecting that. And it's efficient. Efficient means it eliminates contagion. Now, contagion is more of like there's evidence that it could be used prophylactically. So we're really excited about this. And we have an IND in the US, and we're phase 3 ready in India. And we currently have a clinical trial in India. It's a dose optimization study, and I'll get to that in the pipeline. I'll give you some more details and an update on that. So again, stay tuned. Additional indications of prolectin, and we had a case study here, and the case study was on shingles. Look at the before and after picture on the shingles. So what happened is you develop these kind of lesions that are really painful, and you want them to go away as quickly as possible. And we're looking at hours on this chart on the bottom there. And then you'll see here, topical galectin abated the pain in 3 hours. And this was on a case study that was published. So again, I encourage you to go read the case study. It's in a press release of ours. So now, again, all this evidence is to suggest we have one powerful broad-spectrum antiviral platform. So let's move on to our next platform technology of hypoxia. Hypoxia is it's basically oxygen deprivation in your tissues. I'm really kind of dumbing it down. Remember the idea of holding your breath? When you hold you can only hold your breath so long, and there's a reason for that. It creates a hypoxic environment. You need oxygen. So let's move on to the next slide here. BIXT25 is a solution that we have. It's an oxygen bridge. And what you're seeing is these blue molecules are going right through a clot. That's what this graphic is showing you. It's our molecule is 5,000 times smaller than a red blood cell. This is not to scale, otherwise you wouldn't be able to see it. And it's going to be used or expected to be used in ischemic strokes and hemorrhagic strokes. And it's going to reduce the time to needle by 95%. We're going to see that on a follow-on slide, how we do that. So how does it work? Well, basically we took blood. You see the graphic on the left there, blood. We took blood, we pulverized it to the point where we got rid of the globin of hemoglobin, and we were left with the heme molecule. Now, heme is what carries the oxygen. And it needs to be used, so this technology needs to be used via intravenous. It's not like pill, never will be. It's intravenous. It doesn't have any impact with whether your blood type, and is very stable. So now let's get to the use case. The golden hour dilemma. The golden hour dilemma is once you see a person's having a stroke, how quickly can you get them treatment? Because while they're having a stroke, their brain is essentially holding its breath. So how do you get them to the doctor as quickly as possible to figure out if they have an ischemic or a hemorrhagic stroke? I hope I went too far. But anyhow, once you see the symptoms, the ambulance arrives. So our ideal concept is to intervene right at this level. And that's reducing the time to needle by 90%. So, let's reduce this level. Let's give you an oxygen bridge. Have your brain stop holding its breath while we figure out whether it's an ischemic or hemorrhagic stroke. You see, TPA, which is used to treat ischemic strokes, will kill you if you give it to a person having a hemorrhagic stroke, which is why we need to wait before we give treatment. This was a proof of animal concept that we did. I'm going to gloss over this a little bit and focus in on the center item here. So you'll see there in this study, what we did is we hemorrhaged the animals. We took out 90% of their blood. So imagine taking 90% of your blood and living, right? It doesn't seem right. But anyhow, They took him out, they put it in at the same time, a molecule similar to what we have, and the dogs recovered. That's the essence of that article, and you're free to look at it. But that was a proof of concept and why we are so optimistic about our future prospects. It's a big market, and that's all this slide is about. It's a big market, and 500 billion is understating it when you look at the indirect costs. There's limited treatment options. The only option is what I just talked about, is this thing called TPA, and we would go in advance of TPA being delivered. That's the idea behind it. How we do that is, or how are we seeing moving the clinical trials forward? We have some incredible technology, which is a third platform technology that we have. It's that measures tissue oxygenation. Again, not going to get into the specifics of it, but know it exists. And then I see myself running a little short on time, so let me accelerate this a little bit here. This is something that you should know of as a company that we're very interested in, because there's just so much evidence to suggest that we should be going after cancer. But resources don't exist to allow that. So let's just show you why we're so excited about this. The lectins are linked to cancer, and that's our target. And in this particular slide here, in the left-hand side, it basically shows Keytruda was 100% effective in small cell lung cancer if you simply discriminated by tumor type on the people that had high galectins versus low galectins. That's amazing. The people that failed are the ones in red, and they had high galectin expression. So what happens if you block them? You probably turn into the people that are green that had low, right? Because you're lowering it, and they all respond. It was 100% effectiveness of Keytruda. And, you know, no one's talking about that, but we want to. There's also ample studies that were done a number of years ago on galectin antagonists, 140 patients, 9 studies. I mean, but back then we didn't have liquid biopsies. We couldn't prove that we could shrink metastasis. You had to do it with solid tumors. So there's a change in technology. Now here's our pipeline. We have prolactin M, we have prolactin I, and then we have special use cases of prolactin I and ARDS and fibrosis. But where are we at? Well, we're in phase 3 ready. We're in India. We are in a dose optimization. We're a quarter of the way through, so that's some breaking news right now. We're a quarter of the way through recruiting patients. And it's taking a little bit, and we can talk about why it's taking so much, because we developed a really solid trial. That's why, and it's harder to recruit these really good patients that are very sick than what we would have expected. So now I skipped ahead. Let's look at our platform technologies. We have our oxygen carrier. We have prolactin M, which is our galectin antagonist that's going after the glycovirology platform. And then we have this MDSU. It's an FDA-approved device that you saw there, and it's going to help us with drug development with BIXT 25. So there's some synergies there. And then our business strategy. This is very important to pay attention to. So we want to license this technology. So we do our dose optimization trial. So follow along in the flow chart here. Then we get a registrational trial, and then we get approval. Once we get approval, look to the right. That's what a typical deal looks like in India. $30 million and 5% royalty. And we've had some preliminary discussions to try to figure out that's kind of what we're aiming for. So that would give us, so once we license it, that would give us the money to do the next generation platform technologies that all those disease indications go after. Now, let's say that we're working a parallel path, so there's the or button, or we partner with smaller big pharma and we expand the glycovirology very simply. look at all, we can cost effectively go after and do studies on HIV, Ebola, Marsburg, Nipah virus, Epstein-Barr, who knows? We can expand the platform technology really well. And then investment summary here, we have an experienced management team. We've already shown that, right? We have cost-efficient development. AI is helping us move things along dramatically. So we're not spending a lot of money on useless R&D. We've de-risked, I shouldn't say de-risked the science, I'd like to say validated the science with our clinical trials. We're at a value inflection point once our dose optimization comes to pass. And we have institutional investors, loyal shareholder base, all looking at what we're doing. And we have a massive licensing technology. Our symbol's BIXT, we're fully reporting OTC, not a reverse merger. Lots of shareholders, mainly retail, 166 million shares outstanding. Notice the shares held by insiders, a lot of shares held by insiders. Big Pharma is not going to tell us what to do unless they do the right thing. That's a hint there. And then 30 million shares in the float. People like to know that. And a very compressed market cap of 20 million. And that concludes my presentation.
Great job, Mike.
I'm
All
excited.
Right. Good job. That's
you. Something was pressing on you, Anna. What's cooking?
Yes, it was. Well, I just read it in the news. Obviously, we've heard about that bird flu found in dairy cows in Texas, which is pretty close to where I live. They found out the virus was able to jump to humans. I don't want another pandemic. No one does. I don't know how serious this is, but I also heard that the virus is spreading. Over 2 million chickens were infected and died of bird flu in Texas. So these viruses, they could have a major impact on our food supply. So talk a little bit about that, Mike. Do you have any idea if your drugs might be able to treat people, God forbid, if they're infected with this bird flu and the animals?
No, that is a timely question. But I want to explain the real focus of our drugs is human use. So let's let investors know that we're not going to go off on this tangent unless someone bops us on the head. But I'm going to answer your question. So our strategy is to get this drug approved in humans. So if someone wants to see if it works on what's called bird flu is called H5N1. So if someone wants to see if it works on H5N1, which is bird flu, we've already tested it on, remember this, H1N1. The only difference between the two is the 5, right? We think there's probably a pretty good chance we know how to do it. I invite the inquiries. So the thing is, look, we have limited resources, so we're not going to go out there and test for H5N1. But do I think it's probably going to work? I'm not going to say no. The discussions come up.
Okay, well, just in case, good to know. James Baker wants to know, when is the phase 3 trial projected? And will it be in India? And how does the U.S. view trials done in India? Do they require duplication within the U.S.?
So the timelines are so difficult to project right now, but it's an excellent question. And let's answer this by sharing with you what our challenges were. So we dosed our first patient, so we started the dosing. But what's happened is the people in India, what we did is we tried to harmonize with the U.S. market. So we were going after people without underlying medical conditions. And what we're finding is a lot of the people that are coming in and that want our drug have underlying medical conditions. So that makes it just a little bit more challenging. So it's no longer that we're facing no cases of COVID in India. Now we have cases of COVID in India, but we have to find the right person for that trial. So we have to stick to the protocol. But the good news is once the protocol is done, then we'll be able to go to a phase 3. So the answer to your question, Chase, is, I think his name was Chase, right? The answer to your question is, it's too early to give timing until we have funding in place to do the phase 3. But the idea is to get people to understand that money that we get funding for the phase 3, that they understand what's on the other side of that. You saw what was on the other side. A typical deal in India like this is going to be 30 million. So, a couple million to 30 million, I mean, those are the economics that shareholders should be looking at.
Have you had any successful partnerships with any big pharma or small pharma companies?
We are talking, but there's barriers. So it's maybe to be a little bit transparent. It's not as I went on emerging growth and I had this great game plan. I'm going to talk to a bunch of big pharmas out there, and I have been talking to big pharmas. But the hard part is getting over the barrier that you're going to them and you basically, and I hate to use this analogy, but you have to call their baby ugly. And who wants to talk to you when you're calling their baby ugly, meaning their own pet project? So they can't see beyond their own pet project to let you in and be open and receptive to just even hearing your pitch. So I'd say that's probably my biggest barrier with small pharma. And we'll be able to approach big pharma after we get this dose optimization study done, because I think we're going to find out that a smaller number of pills, we don't know this yet, but the theory is a smaller number of pills is going to get the trick done.
Well, Mike, we are out of time. As always, wonderful presentation. Maybe a second of some closing remarks for you.
I came across with a different presentation for all the investors out there. I really wanted to drill home the point that we have an opportunity here to change the landscape of how we look at viruses. And this is the one company, Bioxytran is the one company that I believe holds the key that can unlock this viral threat that we're constantly facing with, like, for example, this H5N1 threat. What about disease X? What about all these other viruses that are out there? We don't know. We don't know yet. So support Bioxytran. Look at the market cap. Look at these valuation models that show where we should be, and do some investor math. Thank you.
Wonderful, Mike. Thank you. We look forward to seeing you again in the near future. Thanks for coming on today.
All right. Bye-bye.
All right, everyone, stay with us. We'll be right back with Alpha 7 Therapeutics.